Hello and welcome to today's lecture of Introduction to Mechanobiology. In the last few lectures, I started discussing about mechanobiology of diseases, and in that context, I had discussed cancer, okay, and shown that in cancer you have this response of fibrosis marked by increased deposition of collagen 1. This leads to ECM stiffening. So, collagen 1 and plus cross linking via enzymes like lysyl oxidase, also represented as LOX. Okay. So, both these things lead to ECM stiffening and reorganization. Okay. By ECM reorganization, where you have ECM fibers form cross-linked bundles which also are aligned. Okay. So, all of these responses in the cell level. So, what we found by discussing the cell paper, we showed that this ECM cross-linking is capable. So, here ECM cross-linking is capable of inducing tumor progression. Okay. So, this leads to, and this tumor progression is via activation of integrin signaling. So, today we will discuss another disease again marked by fibrosis. This is called atherosclerosis. Okay. So, what is atherosclerosis? So, all of you know that you have arteries within our body, okay, and these are blood vessels that carry oxygen rich blood to our organs, to other parts of the body. Okay. So, if you see the cross section of a blood vessel, will have this nice structure, okay, where you have blood flowing through these channels. Okay. So, the cross section is circular. Okay. So, in side view, if I draw, okay, and I have blood flowing through this channel. Okay. Now, what happens in atherosclerosis? The same blood vessel now looks here. You have blood flowing. Okay, the green zone is called an atherosclerotic park. Plaque. Okay. So, this atherosclerotic plaque, essentially it restricts the blood flow. So, instead of flowing through the entire artery, blood flow is restricted. Okay. And this plaque is made up of multiple constituents, including fat, cholesterol, okay, calcium, okay, and other substances. So, over time this plaque becomes, your plaque stiffens and restricts blood flow, eventually causing a heart attack. Okay, where blood flow is completely gone. So, when you do these tests, doctors say that you have 98% blockage in arteries or veins, so on and so forth. Okay. So, though the initiation of this atherosclerosis is attributed to, let us say, circulating factors like LDLs or low density lipoproteins and triglycerides. Okay. So, there are other changes, okay, in the flow pattern, in the way the blood flows through these arteries. And intriguingly, people have observed that atherosclerotic lesions are typically localized, okay, they develop at arterial branches and sites of curvature. Okay. So, what is the reason for that? So, with the help of fluid mechanics, it has been demonstrated that the type of shear stresses at these locations is perturbed. Okay. And this, and when I say is perturbed, so what we have found that these lesions are formed, okay, at locations where shear stresses are low or disturbed. Okay. So, suggesting that the flow profile itself has a role to play in this. Okay. And subsequently, people have shown, okay, that atherosclerosis, okay, occurs mainly in arterial segments, okay, which display endothelial cell dysfunction, okay, endothelial cell dysfunction, okay, triggered by low oscillatory or turbulent, okay, shear stress. Okay. So, let me briefly explain what is this. So, if you take a pipe, okay, if you take a simple pipe, so let us say this pipe is equivalent to our artery, okay, your blood is flowing in a given direction. Okay. So, if you take a small element, okay, so this there is a line of symmetry down the center. Let us take a small fluid element in the center. So, what are the forces that this fluid element is subjected to? So, let us assume that this length is delta x, okay, and this radius. So, because it is a pipe, it is actually symmetric. So, you have this radius r. So, on this wall you have a pressure force P. So, because this is only an elemental length of delta x, which is very small, I am putting a pressure of P plus delta P here, and you have a shear force tau. Okay. So, shear, as you know from I recall from our discussions, that shear is tangential forces. Okay. So, you have these forces, and the force balance equation looks something like that. So, pressure into the cross sectional area is pi r square plus tau into the area. So, because it is the tangential length, tangential area, so 2 pi r is the circumferential length into delta x is the total shear stress, okay, and this must be equal to P plus delta P into pi r square. So, from this equation you can establish a simple relationship like that. Okay. P is r by 2 del P del x. Okay. So, tau is my shear stress, okay, tau is my shear stress. Okay. So, this varies at r by 2 del p del x, which means that tau max must be at the walls. Okay. So, the walls are subjected to maximum amount of shear force, okay. And from this equation you can derive the flow. So, because tau is given by mu du dr, this is Newton's law of viscosity, you can put this equation mu du dr equal to r by 2 del p del x. Okay. You can take you can integrate this equation. So, you can find out that mu into u is, or if I remove mu from this side, is 1 by 2 mu del p del x common, okay, r square by 2, okay. And if I integrate from 0 at the walls to maximum at the center, okay. So, from capital R to small r, okay, or you can integrate from 0 to u, then you would get this equation 1 by 4 mu del p del x, okay, into r square minus r square. Okay. So, minus 1 by 4 mu del p del x is minus, okay, into r square minus r square. Okay. So, you can take out r square common, okay. So, when r is equal to 0, you have u max is, so this is your velocity profile. Okay. Let me write down the equation here, is minus r square by 4 mu into del p del x into 1 minus r by r whole square. Okay. So, there is a minus sign here for the simple reason because del p del x is negative. So, negative into negative is positive. Okay. So, if we plot the velocity profile would be something like this. Okay. So, this is your pipe, this is the direction of flow, you have maximum velocity here. So, this velocity profile is parabolic in nature. Okay. Now, what has been observed is there are two transcriptional factors, YAP and TAZ, okay, yes associated protein and TAZ. Okay. These are highly expressed in atherosclerotic lesions. Okay. So, whether there is any association between YAP TAZ and mechanosensitivity is unknown. Okay. The link is unknown. So, today I will discuss one paper, okay, where they have we have described the flow dependent modulation of a YAP-TAZ pathway, okay, in endothelial cells. Okay. So, for doing this, they use a parallel plate flow chamber. So, where again like a pipe, so you have a parallel plate flow chamber. Okay. So, if you look at the base, you have endothelial cells are grown as a monolayer. And then you have flow. So, this has a given height. So, you have a flow profile being imposed on the cells. So, the cells are subjected to shear stresses. So, using this you can vary the type of flow as steady or laminar. Okay. In laminar flow when you have flow there is no mixing. So, in the pipe if you add a small amount of dye at a given point, the dye will travel along that direction and will not diffuse in another direction. So, this is what is meant by laminar flow. In contrast, you can have turbulent flow where there will be mixing of these particles across this radial distance. Okay. So, what they found was they used UV cells. Okay. This is human umbilical vascular endothelial cells, okay, and then they subjected to laminar or oscillatory flow. So, this is laminar shear stress or oscillatory shear stress. Okay. And what they found was under laminar conditions, so they checked under these conditions what is the profile of YAP and TAZ. Now, these transcription factors exhibit this particular behavior, okay, that when it is inactive, okay, it is inactive in its phosphorylated state. Okay, and it exhibits a cytoplasmic localization. But when it gets active, its phosphorylation levels drop, and leading to a nuclear translocation, nuclear localization. So, when they did this experiment, okay, what they found was under laminar shear stress, okay, their phospho-YAP levels were constant. Okay. So, phospho-YAP to total YAP was constant, okay, but under oscillatory shear stress this decreased, phospho-YAP to total YAP kind of decreased, okay, and under these conditions TAZ, the expression profile of TAZ increased. So, this they observed not only in UV cells, but also in aortic endothelial cells. So, this is aortic endothelial cells and this is vascular endothelial cells. Okay. EC stands for endothelial cells. Okay. In both these cases, they observed this particular profile, suggesting that, so this suggests that OS results in YAP-TAZ activation. Okay. And they also confirmed this by the localization signature. So, what they did was they tracked the nucleus to cytoplasmic intensity signal, fluorescent intensity signal, okay, and then they found was compared to laminar stress conditions where you had a given profile which was lesser than or comparable to 1. Under these conditions this increased in OS, this was significantly increased to a value of 3. Okay. So, this confirmed that YAP-TAZ signaling gets activated. Under oscillatory shear stress. Okay. So, what they then did was they performed gain of function and loss of function experiments. Okay. So, but for loss of function there is siRNA, they knocked down the expression of YAP-TAZ, and for gain of function they overexpressed YAP. Okay. So, this was always constitutively active, and what they found was under these two conditions, okay, when they knocked down YAPTAZ signaling, okay, endothelial cell proliferation dropped significantly. Okay. And when they activated YAP, this led to dropped. Okay. EC proliferation increased significantly. Okay. So, suggesting, okay, so this suggests that YAPTAZ signaling modulates EC proliferation. Okay. And mechanistically what they found, what the percentage of S phase cells, okay, was significantly increased under oscillatory shear stress, okay, as well under when YAP was overexpressed, even under laminar shear stress conditions. Okay. So, together what they concluded was under laminar shear stress, okay, inactivates YAPTAZ signaling, while, okay, oscillatory results in YAPTAZ activation, and this leads to EC proliferation. Further, okay, so similarly what you have under inactivates YAPTAZ and inhibits EC proliferation. Okay. What they then checked was what was the role of YAPTAZ in mediating inflammation. Okay. So, what they found was oscillatory shear stress, it induced expression of VCAM1, vascular cell adhesion molecule 1, and ICAM1, okay, in ECs, okay, and this led to increased attachment of THP1 monocytes, okay, on EC surfaces. Parallelly, when they knock down YAPTAZ with SIYT YAPTAZ, okay, they saw decreased level of VCAM and ICAM expression, leading to reduced adhesion. Okay. Also, when they overexpressed using SAYAP, constitutively active YAP, okay, this increased the inflammatory response, even under static conditions. So, even under laminar shear stress conditions, you had inflammatory response when you overexpress SAYAP as quantified by the number of monocytes which got adhered. Adhesion. Okay. And all of this was made possible, okay, via expression of integrin beta 2. Okay. So, what they found was integrin beta 2 expression, okay, led to beta 2 expression was activated in THP1 cells, okay, and this is the mechanism which explained the enhanced adhesion of THP1 cells onto the ECs. Okay. So, together what the authors concluded was that YAPTAZ activation by oscillatory stress, okay, these enhance monocyte EC interaction, okay, and these can contribute to atherogenesis. Okay, this can initiate atherosclerosis. Okay. So, together the authors came to the following conclusion. Okay. So, whenever you have laminar flow, okay, your YAPTAZ exhibits a cytoplasmic localization, okay, and as a consequence, okay, there is the overall situation is atheroprotective, but under disturbed flow, okay, YAPTAZ phosphorylation goes down, that leads to nuclear localization, upregulation of VCAM and ICAM expression in ECs, and integrin beta 2 expression in these cells. So, overall this leads to proliferation and a inflammation signal. Okay. So, this shows you that other than soluble factors, okay, you can have this mechanical flow induced inflammation created in zones where flow gets disturbed in either one direction or where the shear stress is low, that leads to build up of these atherosclerotic plaques, or if the flow is disturbed in either direction in oscillatory, then also you can get these zones. Okay. With that, I thank you for your attention.